Hello to all of you. I'm going to give you the instructions for the lab, Dahnia lab. So basically, this is the Dahnia lab. Basically, what we are trying to do in this lab is it's basically a scientific method lab. So I'm not going to go into the details of the scientific method because you already know. We have already discussed. You have already discussed in your theory classes what scientific method is. What are the steps of the scientific method? So basically. This is a scientific method lab. In this lab, the aim of this lab is to see, to examine or to check the effects of alcohol on the heart rate of Dahnia magna. So Dahnia magna is a aquatic organism, and something very important about this organism is it's transparent, and it's a transparent organism, and you can see the heart of this organism very clearly under the microscope. So I have already posted 2 videos for you guys, so please check those videos and see how this organism looks and how does you can detect the heartbeat of this organism. So um, basically, the only thing I would like to add in the scientific method is the hypothesis. There are two kinds of hypothesis. One is a null hypothesis, another is an alternative hypothesis. So null hypothesis is that hypothesis which says that there is no relationship between the two variables you're trying to study in an experiment, and it's always opposite to what we're trying to study. And the alternative hypothesis is a hypothesis which says that there is a relationship between the two variables. So basically, the steps of scientific method are, we observe something, the question comes to mind, then we formulate a hypothesis, then we perform an experiment, and then we get the data, and then on the basis of the data, we make a conclusion. So these are the steps of the scientific method. There are two kinds of variables. One is a dependent variable, another is an independent variable. So independent variable is that variable which is in the hand of the scientist or a researcher. That's what you control. And the dependent variable is the variable which changes in response to the independent variable. So this is the picture of Dahnia, and you can watch those videos. So basically, if we would have done this lab in the laboratory, so what you would have done, I'm going to just talk about the steps. So you would have taken the Dahnia and you would have put them in the on the slide, and you would have first of all observed the Dahnia, you would have focused the Dahnia, you would have seen the heart beating in the Dahnia. So then you would have put the 2 drops of the pond and have checked out the heartbeat of this. You would have counted the heartbeat of this organism. So because the pond water does not have any alcohol, so this will be our control. So it does not have any alcohol. Pond water does not have any alcohol, so it will be like our control. So this is our control group. So you would have checked or counted the heart for 15 seconds, and you would have written here, and then you would have multiplied by 4. That would have given you beats per minute. So for every group you would have done 5 trials, which means you would have done it 5 times. So you would have counted the heartbeat for 15 seconds, and then you would have given the rest for 45 seconds. Then you would have started again counting the heartbeat for 15 seconds, and then you would have rested for 45 seconds. So you would have done 5 trials. Okay, so this is our control group. And then what you would have done, you would have removed the pond water, and then you would have added 4% ethanol. So we're trying to see what we are in this experiment, which is a scientific method experiment. We are trying to study what is the effect of alcohol on the heart rate of the Dahnia. So your null hypothesis will say that there is no effect of alcohol on the heart rate, and alternative hypothesis will say there is an effect of the alcohol on the heart rate of the Dahnia magna. So you would have replaced the pond water then with the 4% ethanol, and then again you would have done the same thing. You would have done this thing 5 times, and you would have done 5 trials. So in every trial you would have counted the heartbeat for 15 seconds, and then you would have waited for 45 seconds. So whatever the heartbeat you get in 15 seconds, you would have you will multiply by 4, and then you get the beat in. That's you multiply by 4, you get the beat in a minute, and this is going to be done 5 times. And similarly after that, so this is your experimental group. You would have replaced 4% ethanol with 6% ethanol, and you will be doing the same thing here also as you did earlier. And then you are going to, so you have three groups: control group, 4% ethanol and 6% ethanol, and every for every group you have done 5 trials. So you take the average and then you would have put it here. So this is the data from your group, and then in the lab there are different groups, so you would have taken the data from other groups, you would have written here. So you would be writing here the data for control group for 4%, from 6% for group number 1, for group number 2, group number 3. So you will be writing all the data here, and then you are going to take the average of the whole class, all the groups, and you would have written them here. Okay, and then basically then you were supposed to make a graph. So so you would have made the graph, this data from the whole class. So for this, basically when we make a graph, we have two axes, one is the x-axis, another is a y-axis. So on the x-axis is our independent variable, which is the percentage of the alcohol you have used, and on the y-axis goes the heartbeat or the heart rate per minute of the Dahnia. So on the x-axis always go the independent variable, and on the y-axis always go the dependent variable. So that's what you would have done in this lab. So and I have provided you with this data. This is the data. So this is a data which is not going to go and fit into your lab manual, because I am providing you with this data so that you could see that what kind of results you would have got if you would have performed the experiment in the lab. So if you see here, this is the data from different groups. This is like section 8, section 9, section 10, section 11, and these are the different groups in these sections, and you can, these are all average values written here. So so basically, like if the in the control group, if the heart rate is 232, this is in the control group. With the 4% it becomes 186, which means it has decreased, and with the 6% it becomes 158. So you can go over and check other data also. What you will see, like here it is 190, it becomes 116, and with 6% becomes 60. So basically why I'm providing you with this data is I want you to go over and check this data and see that as we are increasing the concentration of the alcohol, the heart rate of Dahnia is actually decreasing. So that is what is the aim of the experiment. We wanted to see what is the effect of alcohol on the heart rate of the Dahnia. So I'm not asking you to take this data and put in the worksheet, and I'm not even asking you to complete the worksheet which is given here. I want you to go over this lab. I want you to understand this experiment and have a look at the data, and what I want you to do is I want you to go on D12, and I have created a quiz for you there. After you have gone through this lab, please take that quiz, and that quiz is a graded quiz. That's all I want you to do. I do not want you to complete all the questions in this lab, because I have created an assignment for you which substitutes all these questions which are there in the lab manual. Okay, so if you have any question, please let me know.